Hello everyone, my name is George Georgiou. I am a curator here at EMBL-EBI, part of the Gene Ontology Annotation Project, and I'm here today to tell you how to use QuickGO and GO slims for your research. To start off, let's first talk about what GO is. What GO stands for is the gene ontology, and what this is a way to capture biological knowledge in a written and computable form. Now, what this actually means really is that it's a controlled vocabulary that allows us to use the same vocabulary to describe a gene product, which refers to anything that is either a protein, an RNA, or a protein complex, in the same terms between species, allowing us to make them comparable. And these vocabulary terms or concepts are actually arranged in a hierarchy, and they're related to each other through, they're all connected to each other through a series of relationships. Now, what you'll notice next to the text here is an ancestry tree that we have explaining the GO. So at the top of it, we have less specific concepts. So for example, we have biological process, which really is a very generic term. And the further you go down in the tree, you get to more specific concepts like the negative regulation of signal transduction. And you'll notice these different colored arrows connecting them all. And these are the different relationships that connect GO terms together, this controlled vocabulary. I won't go too much into the relationships today. I've done this in a previous QuickGO video that is available on YouTube, which if you haven't watched, I strongly suggest that you do after today. It's worth a read to understand the new functionalities that QuickGO can provide, but I will be going over some of the usage today. So the gene ontology is actually three structured ontologies, allowing us to describe a gene product in terms of the biological process they're involved in, their molecular function, and their cellular components. Now, what this means is that we describe a gene product for the process that it's involved in. So let's say we have a kinase, and the kinase would be involved in the biological process of protein phosphorylation. It would have the molecular function of kinase activity, and the cellular component is actually where the gene product has this activity for this biological process. So we'll say it's in the cytosol. Now, to get to the crux of why we're all here today, how bench scientists like you, or even non-bench scientists who are interested in getting into this, can use the gene ontology. And here are some of the case studies that we'll be going over. The first one will be to access gene product functional information, obtain functional information for novel gene products, validation of experimental techniques, and analyze high-throughput genomic or proteomic datasets. Now, where can we view all this? This is going to be on QuickGO, the web-based browser that we host here at EMBL-EBI for viewing and browsing the GO annotations. So this is the QuickGO webpage, homepage. The link is below. If you have a chance and you'd like to click along, I've structured this webinar today to work like an interactive seminar where I provide instructions for you to kind of follow along with what I'm doing so you can actually explore the functionalities and how to use QuickGO. Now, at the home page, you're probably wondering now where can I actually see the GO annotations? And what I'd like you to do is go ahead and click on View GO Annotations. And this is where we're going to be able to view all our GO annotations and filter them. So you'll probably see a table like this in front of you. And most importantly, as you're looking at it, it probably doesn't make much sense. So one thing we're going to go over right now to make this more practical for you is actually how to read a GO annotation on QuickGO. Starting with the columns, gene product and symbol. It just lists the database that we're using with the identifier for that database, and the symbol is the gene product name, because if you notice, like for the uniproduct session ID, the symbol CIB4 is a lot simpler to remember than A0PJX0. So it's just a quick way of understanding which gene product you're looking at. The next column is what we call the qualifier, and this determines the relation of the gene product to the GO term. For cellular components, we use the qualifier part of. For molecular function, we use the qualifier enables. And for biological process, which is all the way down here at the bottom with O-glycan processing, we use the qualifier involved in. In addition to these, we actually can modify the interpretation of an annotation with other qualifiers, co-localizes with, and contributes to. Co-localizes with is for cellular components, and it's to describe a gene product that may actually not be part of, let's say, the Golgi complex, but co-localizes with it. So this is how we kind of enhance the information that we can capture with this controlled vocabulary. Contributes to is for molecular function, and we use this in the case of sometimes saying a protein has a function when it's part of a complex but doesn't have the function on its own. But when it's part of that complex, the complex is active. So we say it contributes to that molecular function. And finally, you see one here that says not. Not applies to every qualifier, including the enables, part of, and involved in. And what this is that if we have a case, for example, where we have one protein, one isoform is catalytically active, so it has this, it enables this one molecular function. So we'll say, again, with the case of kinases, we have an isoform that is catalytically active, so it has kinase activity. But another isoform is missing a catalytic residue. And in order to distinguish this, what we would use is a not qualifier. That way, when people looked at it, they would see, okay, isoform 1 has catalytic activity, but isoform 2 does not. Next, we have the GO term. So this column actually lists what the GO term ID is and the aspect of the ontology it represents. So you'll notice there's a little letter C for cellular component, F for molecular function, and P for biological process. In blue is the GO term ID. If you click on that, it will actually take you to the definition page for that GO term. Underneath that in black is the actual GO term itself. And next to it is a basket if you wanted to put this in the basket for QuickGO. And if you want to see where this falls in the ontology, there's these little blue trees over here. If you click on that, it'll bring up a pop-up of the gene ontology tree and show you where that particular GO term falls for that particular aspect. Annotations are supported by evidence. We have to provide an evidence code. So in this case, we've traditionally used GO evidence codes, what map to evidence code ontology or eco codes. We allow you to do the same thing that we do with GO terms. If you click on the little blue tree, it shows you where it falls in the evidence code ontology branch. And also, if you click on the eco code itself, it gives you the definition. Now, evidences are getting more and more specific. We are switching over to eco more often, which allow you to describe direct assays. And to give you an idea of the evidence codes that we have used historically and how they're going to be mapping to eco, these are the evidence codes that we used to use in GO curation. They all map up to eco terms. So we have things like inferred from direct assay, inferred from mutant phenotype, inferred from protein interaction. These are all ones that we commonly use. And the ones below, like ISS, TAS, NAS, these are ones that were not commonly used. But to give you an idea of what is an IDA or an inferred from direct assay, we consider these enzyme assays, in vitro reconstitution, immunofluorescence, cell fractionation, and some of the ones that we don't commonly use, like traceable author statement. It's usually when we read something in a paper, we can then say we see that an author has cited a specific paper for making a statement, so we'll go ahead and capture that. But these terms over in the lighter shade of green on the bottom half, we don't use as often. And they all map up to an evidence code ontology, which can actually be more specific to the experiment that was used to support that GO annotation. Obviously, with evidence, we have to provide a reference. We have to say where we found this information. And this really pertains particularly to manual annotations, so things that have these evidence codes like IDA and IMP and IPI. And in QuickGO, we allow, when we list the reference, you can click on that reference and it'll take you to the EuroPMC page where you can actually view the paper that we got it from. So if you want to read the paper yourself, you can. The with from column is actually really specific to the evidence codes IPI and IGI, so physical interaction and genetic interaction. It allows us to list the interacting gene product, or if a gene product is interacting with a metabolite or an ion or a drug in particular, we can use the proper identifier in the with from. So when people read the annotation that CYP4 has protein binding with the unit product identifier P08514. Taxon tells us what the gene products, the species of gene product comes from. The assigned by lists the group that created the annotation. So for this slide here, I've only selected from Uniproc because that's who we work most closely with here at EMBL-EBI. But there are other groups that do contribute GO annotations. There's an entire consortium, and if you're interested in that, you can go to geneontology.org. And finally, the annotation extension. What the annotation extension does, it actually extends the GO term to make a more specific GO term. So GO terms, as we said, have to be usable across all species that we study in biology. And an annotation extension allows us to combine Go terms with other ontologies, like cell line ontology and Uberon ontology, database IDs. So if we wanted to say that a particular protein binds a particular segment of DNA, we can use an ensemble identifier, and in addition to Go terms. So if a particular molecular function happens only during a part of cell cycle, we can capture that in the annotation extension. So in this case that we have up here, we see that SLC5A10 is part of a cell line, and it's an extracellular exosome that is made by this particular cell line over here. And if you click on it, it will actually show you what the cell line is. And if you zoom out a little bit, now we're taking a look at the entire page. At the top, you see these blue tabs highlighted here by the red circle. These are our filters, and we're going to go over how to use those in a few minutes because that's actually how we're going to find a lot of our information today. So let's get into our first case study. Access gene product functional information. So if you're on the website now, this is great. You can follow along. So in this case, you don't have to follow with my case example, but you can use your own. Let's pick a protein and get the UniProt identifier for it, or you can search for it by typing in the name of it in the search bar in QuickGO. But for the sake of speed and time, I've already chosen one, which is human CSARC. It says UniProt ID P12931. This is a protein I'm familiar with from my own PhD work, so I figured it was the one that I could talk best about. So why don't we go ahead and follow these instructions here, which is to take that UniProt identifier, put it into the search bar, and then we're going to go ahead and walk through the steps to get to the annotations for it. So I'm going to want to filter for manual experimental annotations, and I'll explain what these are in a second, and filter once more for annotations made by UniProt. I'm only interested in UniProt annotations for this example. So here's the search bar again in the front of the home page. If you pop in the identifier, you should see a little pop-up appear, and it'll show you in the gene product section the proto-oncogene tyrosine kinase SARC with 504 annotations. What I'd like you to do is click on Show All One Result. And it'll bring you to a page like this, where you have the results on the right-hand side and on the left-hand side, a filter that you can use to, if you had more than one hit, and this gene product search would be typed in, let's say, by name rather than UniProt identifier. And what I'd like you to do next is to go ahead and click on the 504 annotations, please. So now you should see this annotation table with the gene product ID filter highlighted, meaning that it's filtered for that particular UniProt identifier, and we have 504 annotations. But this includes electronic annotations, which actually make up the majority of Go annotation and represent the majority annotations we have for a majority of species. The reason being is that we don't have enough published data on every single species, and one thing that QuickGO does is it provides electronic annotations to all the available species that we have in UniProt. But in this case, I'm interested in manual experimental annotations, so ones that come from papers and that have been manually curated by curators like myself. So if you were to click on the gene product ID, you'd see the SARC UniProt identifier there. If you actually wanted to, we could have skipped the search bar, gone to View Go Annotations, popped this filter open, typed in our UniProt identifier in the field there, then clicked Add, and then hit Apply, and we would have ended up at the same display. So let's go ahead and filter our annotations from manual experimental evidence. What I'd like you to do is click on Evidence, scroll down till you see all manual experiment codes, select that, and click Apply. Finally, if you click on the More tab, you see additional filters that weren't present before. These include qualifier, reference, with, from, and assigned by. As I said before, I only want UniProt annotations for this example. So I go over to the assigned by column and I'll scroll down to UniProt. They're in alphabetical order, and each one of these groups that's listed there, you can always filter for the annotations from that particular group if you're interested in that one. But in this case, as I said, we only want UniProt. When you're done, click Apply. So here's our filter results. We got 56 annotations from UniProt, all with different Go terms, well, some of the same with protein binding. Our evidence codes are manual experimental ones that we've placed in. We see the references, and with the protein binding, we see interactions for human SARC. And we even have some annotation extensions that we could look at. But in this case, I would just like you to take a look at the Statistics tab, because this is actually kind of interesting, where you can find out a brief summary of everything that we filtered. If you click there, you'll get a display that looks like this, and it'll tell you you've pulled out 56 annotations to one distinct gene product. Obviously, if you were to put in multiple gene products, you would have a higher number of distinct gene products and probably a greater number of annotations as well. The next tab is the Go ID. This gives you a summary of the annotations per Go ID. So you'll see how many annotations have been made to protein binding, to protein tyrosine kinase activity, and then also you'll see gene products per Go ID. If you had more than one gene product, obviously it'd be higher than one. Aspect will tell you the rough percentage of the breakdown of the different ontology, the usage of different ontologies for the annotations that we filtered for. So for example, we see molecular function makes up the majority at 48.21% or 27 annotations. And gene products per aspect, again, it's because we only have one gene product that we filter for, will be one. Evidence code, it'll actually break down the frequency of an evidence code. So ECO 353, if you click on that, it will say it's inferred from physical interaction. 315 would be mutant phenotype, and 314 would be direct assay. If you were to then click on reference, it would list off the PMIDs that were used to make these annotations and how many annotations were made to that individual paper. Taxon will be the species that we use. So in this case, it's going to be human, so 9606 is a taxon ID for that. And the assigned by, because we filter for UniProt, will only show UniProt there. So this is just one area that you can actually take a quick look at if you didn't use any filters, to see what the breakdown is and which database gave you the most, or which Go ID is used the most in the ones you filtered, or which reference was provided the most annotations. So that's one way that we can use QuickGO. Now let's take a look at our second case study, which is to obtain functional information for novel gene products. Let's say you have a grant coming up and you're not sure what you want to study, but you know you want to look at something that's going to be a popular topic, or something that's not going to be very well studied, and you just want to see where the knowledge is a bit lacking for some things. So for this case study, I said DNA polymerase because DNA repair has always been a hot topic. So we're going to use the search bar to search for DNA polymerase. We're going to select the results for the gene products, and then we're going to pick a particular species. In this case, we took Bacillus cereus, and we filtered this from the organism column on the left-hand side, and we're going to take a look at DNA polymerase 1 from that list. So go back to the QuickGO homepage, type in DNA polymerase in your search. You'll see that we get hits for terms and hits for gene products. Hits for terms will take you to the Go terms that relate to DNA polymerase, and then gene products will only give you the number of gene products that actually, the gene products actually have annotations. So I want you to click on the 2 million some odd results, please. Don't worry, we're not going to read them all. Now, this page you saw before is a lot more filled now from the results for searching for this. What I'd like you to do is to use the left-hand side, go down to organism, and filter for Bacillus cereus. After that, the page should look more like this. Now we see that we have a more reasonable number than almost 3 million gene products. We now have almost 7,000. But we're going to choose the first one that has more than one annotation. In this case, it's DNA polymerase 1 from Bacillus cereus, the one with 25 annotations that I've highlighted. I'd like you to click on the 25 annotations, please. And this brings us to this table. Now, you'll notice one thing right off the bat is that all the evidence codes are electronic annotation, IEA. And the reason for this is that there are proteins in UniProt that have no manual annotation. Electronic annotations are made using sequence homology from other proteins that we do have information for to give us an idea of what these genes may do. So for this case study where we said we're looking up a novel gene product to use for a grant, we've taken a look at a DNA polymerase that is from a species that's not very well studied according to Go annotation, and we only can guess based on sequence that it has these activities. So this could be an interesting one to look a bit into, and it's not definitely an end-all be-all to finding your next protein to work on, but it does give you a start. Next, let's talk about validating experimental techniques using QuickGO. for this case study, what we're doing is we're going to pretend we've done a cell fractionation using ultracentrifugation and mass spec. And we've got a list of proteins. We want to make sure that the proteins that we separated in each compartment are in the right compartment. So I've just taken some easy examples: human GAPDH, Abel kinase, and glycogen synthase kinase 3 beta. And we only want to see annotations that come from literature because we're trying to validate an experiment. So we want to make sure that these things have been seen where they're supposed to have been seen before in the literature. This is not always the case, but for this example, this is what we're going to do. So I'm going to want to use the QuickGO to filter for annotations to those uniprot identifiers listed there. So then filter for the respective aspect. In this case, we're looking at a cellular compartment, so we're going to want cellular component. Then filter for manual experimental evidences, because I said we want things from papers that are experimentally backed. And then also, finally, we're going to also filter for PMID references. So PMIDs are PubMed IDs. So back to the home page, click on View Go Annotations. Should bring you back to our annotation table. I want you to click on Gene Product ID and pop in those 3 IDs up there. Click Add and then hit Apply. Once you've done that, you should see that we have 669 annotations. Click on Aspect, then select Cellular Component and hit Apply. Now we're going to filter for evidence. Like we did before, we're going to scroll down, we're going to select all manual experimental codes, and we're going to hit Apply. Now we're going to go into the More filter, go to Reference, and scroll down for PMID. Once you've selected that, hit Apply, and let's go back to our annotation table and take a look at the results. So now we have 43 annotations with annotations that are backed by manual curation as well as papers. So we see that most of these papers are most of these proteins are either found in the nucleus or the cytoplasm. And if you click on the PMID, it'll take you to the paper that actually provided that evidence. Actually, a really funny thing that you can do with QuickGO in a way is that if you want to get a quick summary of a paper, for example, and you knew the PMID ID, or you see that this PMID is being used to make this annotation to this one gene product, but you want to see what else was in there, and you want to get a quick summary, take the PMID ID, go to the More filter. So if we go back a slide, if you go back to the More and to the Reference, if you click on the Add More section, this over here, you can just copy and paste your PMID ID in there, hit Add, and then Apply, and it'll actually show you the annotations to that particular paper. So it's actually a nice way to sum up, get a quick summary of papers using gene ontology. Alternatively, you can also do this same task with a GoSlim, but I'm going to discuss GoSlims for another feature, but you can always try this yourself afterwards if you'd like. So finally, the last case study we have is to analyze high-throughput genomic or proteomic data sets. But to do this, actually, I kind of have to explain what a GoSlim is. And I did a little bit of this in the last webinar, but I'm going to go ahead and review this for you. GoSlims are cut-down versions of the Go ontologies containing a subset of the terms in the whole Go. In other words, we are just cutting down a lot of the specific terms for these really generic terms that will kind of give us an overview of the proteome that we're looking at, so the things that have been annotated to it. And these are really helpful when you're doing big experiments like a microarray or cDNA collection, or you're trying to analyze mass spec results and you get a lot of hits. And on top of that, you can either use a predefined GoSlim or make your own. But these are always on a case-by-case basis when you make your own, because you have a particular area that you're interested in looking at, or you have a particular species you're interested in and you only want to look at things that are relevant to that species. So for this case study, we're going to pretend we did a proteomics experiment. This is where that Word document in the material section comes handy. If you can open that up, go ahead and highlight all 25 of those identifiers and just get them onto your clipboard real quick. So we've done an IP from our protein of interest. We don't know what it interacts with. It's a newly discovered protein. We're going to call it protein X. It's from a mouse cell line, and we did the IP of protein X, and we sent it to the mass spec to see what it may interact, what came down with it in the IP. And I know this is a really low number for mass spec. Typically, it's in the thousands. But for this example, and for the sake of our sanity, we're only going to look at 25 proteins that we found as potential interactors for protein X. And we want to know what their biological role is, because this can give us a rough idea of maybe the things that protein X is involved in. For this example, we're going to use the predefined mouse GoSlim. So GoSlim_mouse. And we're going to use, when we've highlighted that, I want you to click on Add Terms to Selection. We're going to copy and paste the UniProt IDs into the gene product ID field, and then hit Add to Selection. And then we're going to apply, and then go take a look at our results. So how do we set them up? Go back to the QuickGO homepage, and I want you to click on Explore Biology. This is where we can view, select, and create our GoSlims. Select the mouse GoSlim from the predefined list and click on Add Terms to Selection. What you should see afterwards is something like this, where you see the 45 terms have been selected, and if you scroll up and through it, you'll see what terms have been added for each aspect of the ontology, so the biological process, molecular function, and cellular component. On the right-hand side, where it says Additional Options, you'll see Gene Product ID. If you can click on that and go ahead and paste in those identifiers that we got from the Word document and click Add to Selection, when you do that, you should see this word, see how many gene products have been added. It says 25 gene products. That's when you know you've done the right thing and it's working. After that, please click Apply. So we get something that looks like this. This is our GoSlim results from our proteomic analysis. So we see two new columns here, one that says Go term and the other one, original Go term. Go term refers to the Go term that we use in the slim. So on the previous page, that mouse slim, these are all the terms that are there that we use. And the original Go term is a Go term in the annotation that maps up to the slim term. So to give you an idea of what this means, for example, you see SEMA3A enables response to stimulants and receptor binding, but it only leads to one Go term, which is chemorepellent activity. If you were to click on the little tree next to chemorepellent activity, it'd bring up the ancestry tree. And this may be difficult for some of you to view, and I apologize. It's a rather unfortunate example that I chose, but it was a bit late to change it. But if you look at where it is on the ontology branch and follow it up, you can actually see that it goes to receptor binding as well as response to stimulus, which is all the way up here. There we go. So you'll have terms that map up to two Go terms. You'll have annotations that slim up to two Go terms, but it's only one annotation. So we see that our protein's involved in a number of different things besides receptor binding as you scroll through the results. And this is actually a rather curious case. And the way to kind of get an overview of it is to go to the stats page. If you go to statistics, you'll see a new little tab that's there. It's called Slim Summary. If you click on Slim Summary, it will give you the annotations per slim term. So the terms that we use to create our slim from the predefined mouse slim, we've now counted the number of annotations to each of these terms. And if you scroll through, we'll see that we have some annotations to protein metabolic process, synapse, cell projection, all kinds of random things that we've chosen. Well, not really random, but our gene products are actually rather random for the most part. I'm not sure if our mass spec results will always look like this. I'm pretty sure if they did, we'd be a little bit worried for the number of varied processes that it's involved in. But for this example, I think it works well. And we're going to be improving this page soon, where you can actually see the number of gene products that it actually has been added per Go term. Now, I don't have enough time today to actually discuss how to create your own GoSlim. So I'm going to give you some tips that were actually courtesy of Palmbase, a collaborator of ours in the Go consortium, that they use to create their own. And I can discuss these with you as much as you like afterwards in the questions. So tips for creating a GoSlim. The first one, when creating a slim for the entire genome, you should try to make sure that it covers as many annotated genes in your set as possible. So if you know, for example, that your species has 200 genes, let's say it's a really, really unusual organism that only has 200 genes, and only 100 are annotated, and when you do your GoSlim, only 100 show up and not a full 200, it kind of helps you figure out statistically what coverage you have. For displaying GoSlims, and also for your own sanity when you're reporting the results of a GoSlim, try to keep the number terms as small as possible to convey your results. The one thing that you should be aware of is that the terms have to be specific enough to capture biologically relevant information. So, for example, many terms like metabolic process or cellular process are too general for the purpose of most SLIM-based analysis. So you want to go something with a little bit more specific, like with metabolic process, you want to look at, oh, I don't know, protein ubiquitination, and a cellular process, apoptosis, but maybe you want apoptosis related to caspase signaling. You have to think about what kind of terms you're going to use and how generic they are and how specific they are. You don't want to go too unspecific, and you don't want to go too specific. So on a related note, if you're using your SLIM for data analysis to summarize, like let's say you did term enrichment, you should ensure that the terms are specific enough to demonstrate the relevance of the biological topic of interest. So for example, PalmBase gives us the example of lumping all genes involved in transport. This may mask the overrepresentation of transmembrane transport versus the underrepresentation of physical mediated transport in your results set. So you need to actually make sure that your SLIM categories represent your results effectively. So just putting in transport may not be good enough for what you're interested in, so you have to put in the types of transport. But at the same time, you don't want to use a type of transport that covers up another. And the way to kind of explore this is actually look in the ontology for these terms. And finally, if you're using an external software that's not QuickGO, like Cytoscape, which can create GoSlims, part of the relationships that establish the connect Go terms, one of them is regulates. And it does exactly what you mean. It means a gene product regulates this biological process or this molecular activity. And it will include that by default, so you'll sometimes get masking of your gene product. So when you're looking at, let's say, regulation of cytokinesis, when you are looking for sets of genes that are annotated cytokinesis. And if you want to know more about these relationships, you can check out our previous YouTube video discussing the gene ontology. So this is something that's a little bit more advanced and is something you're more concerned with. But again, if you have questions, please feel free to ask. And of course, if you need help or would like to provide feedback, if you go back to the QuickGO homepage, we have a help and contacts page. Help is part of our FAQs. Our contact page is where you can reach us. And of course, if you have any questions or comments you'd like to feed back to us, please do. To remind you, our website is ebi.ac.uk/QuickGO. Our email, if you want to get in touch with us, is goa@ebi.ac.uk. If you want us to know some feedback from this presentation, have a question about Go and what you can do with QuickGO, or just curation in Go, or you want us to even curate your paper that you're going to publish, send us an email. We're happy to take a look. And we're now on Twitter, so if you want to keep up to date with what we're up to, please follow us on Twitter at QuickGO_EBI. And furthermore, if you have any questions about, if you're curious if we have more videos, please check out the EMBL-EBI YouTube channel and search for QuickGO. In addition to this webinar, there will be other ones available from EBI, so please check out the training page. And then, of course, if you can provide feedback on this course, it will be greatly appreciated so we know how to tailor the next one. Right, guys, if that's everything, it's been wonderful hosting and teaching you today about our QuickGO services. Take care, everyone. Thank you.